Welcome to the second part of protein synthesis. This video will be covering translation. Here's the key for this video, and then feel free to pause and take another look at it. In translation, this is where the protein will be synthesized based on a decoded gene. There are three main steps of translation: initiation, elongation, and termination. Translation begins when the mRNA exits through the nuclear pores and into the cytoplasm. The 5' cap leads the mRNA out. Initiation, the first of three steps, begins when the mRNA binds to the binding site of the ribosome. The ribosome comprises of a large and small subunit that comes together once the mRNA binds to the ribosome. The energy for assembling all the parts together is through GTP hydrolysis. Let's take a closer look at what's going on.
Okay, so the ribosome is composed of three sites: the E site, the P site, and the A site. The P site is the area that holds the tRNA with the growing polypeptide chain. The A site holds the next coded tRNA and amino acid in the sequence. And the E site is the exit site for where the finished tRNA leaves the ribosome. The tRNA and mRNA move in a right-to-left direction in the ribosome, with the 5' cap leading. The little foam pieces around the ribosome represent rRNA. The ribosome is composed of rRNAs that help build the polypeptide chain by reading the order for amino acids and helping link them together. The ribosome reads mRNA through codons, which are three nucleotides grouped together that code for an amino acid. And each codon codes for a specific amino acid, with some amino acids being coded by multiple codons. Using a codon chart, we can tell what each codon will code for.
The rRNA of the ribosome reads the start codon of the mRNA, AUG, which signals for tRNA to bring the amino acid methionine. tRNA is the carrier of amino acids based on its anticodon side. It gets its amino acids from the cytoplasm of the cell. The anticodon side provides the complementary nucleotide triplet base to pair with the mRNA codons. In this case, for the starting codon, AUG, the anticodon has UAC. Once the starting tRNA has binded at the P site with the first amino acid, elongation can now begin. Elongation, the second step in translation, starts with the anticodon of the next tRNA binding to the corresponding mRNA codon sequence at the A site of the ribosome. The tRNA holds the next coded amino acid.
The rRNA in the ribosome forms a peptide bond between the growing polypeptide chain at the P site and the newly added amino acid at the A site. And the polypeptide chain will shift to the tRNA located at the A site. As you can see, the entire mRNA and its attached tRNA shift down one site for the next tRNA and amino acid. The tRNA without its amino acid at the E site exits the ribosome to pick up another amino acid to repeat this process again. These steps repeat over and over until the mRNA reaches the stop codon. At the stop codon, the final of the three steps in translation begins, which is termination. The stop codon signals for the release factor of the protein. To be specific, the stop codon hydrolyzes the bond between the final tRNA and the finished polypeptide chain. Okay, so this is the end of translation, and the protein is almost complete. The completed polypeptide chain goes through protein processing, where it is folded and assembled into its final 3D protein shape. Thanks for watching our video!